This is Origin Stories, the Leakey Foundation podcast. I'm Meredith Johnson. It's December, and that means it's time for our annual look back at the year in human evolution research. And because the Leakey Foundation is a donor-supported nonprofit, December also means that before we get into today's episode, I'd like to ask for your support. Through December 31st, your one-time or monthly tax-deductible donation will be quadruple matched. So please go to leakeyfoundation.org/originstories or click the link in your show notes right now to support this show and the science we talk about. Thank you so much. Okay, now on with the episode. Origin Stories producer Ray Pang is here with me, and he's going to surprise us with 3 of the most exciting human origins discoveries of 2024. Hello, Ray.
Hi, Meredith. I'm so happy to be here.
It's great to see you as always. So tell me, Ray, what have you been up to?
Well, to get ready for this episode, I talked to 3 different Leakey grantees who brought me their favorite discoveries of the year.
So Ray really does keep this a tight secret. I have no idea who he spoke with or what discoveries they shared, so I'm very eager to get into it.
Well, the wait is over. First up, we have Christian Capelli, a molecular anthropologist from the University of Parma in Italy. He's a Leakey grantee, and his research focuses on the molecular side of human evolutionary history. And he's also doing incredible work looking at the genetics of geladas in Ethiopia.
I love them so much. I think they have the best hair.
Yeah, they're really beautiful animals. He described them as if a baboon got crossed with a cow and a lion.
That's a very good description of them, yes.
But today, he brought us an incredible finding looking at the genetic evidence that has emerged to show us how our ancestors lost their tails.
So first of all, it's not just humans that don't have a tail. It's chimpanzees, it's gorilla, orangutan, even further away, the gibbons and siamang. All of these guys, they all share a common ancestor, and all these guys don't have a tail. So the lack of a tail emerged. It must be in between the point where we share all a common ancestor, which is basically around 20 million years ago.
Okay, so in this study, scientists were able to identify a single gene called TBXT, which is involved in tail expression. It's important to note that this is quite rare because usually when we're looking into how phenotypes are expressed or like physical things on our bodies or other bodies, there are hundreds of genes that are involved in making up one physical characteristic. There's not one gene that determines how tall you are. Like, it's not just tall or short. It's not like an on and off switch. There's hundreds of them. And so for them to find one gene that is responsible for how something is physically expressed in our bodies, it's pretty odd.
It is finally a very neat and clear trait that everyone is aware that we use in our slides to describe what elements that identify apes. And finally, you can tell to the students, well, actually, we now have a very good explanation why all these guys don't have a tail.
Is it common in this kind of work to get such a clean answer?
No. I mean, so we are now in a moment in research where, especially in computational biology, where there is a lot of data, it's not very difficult to generate data. And the complication is analyzing the data. But nevertheless, when you have this large amount of sequences, you can make a lot of comparison. Think about that they found tens of thousands of differences when they were comparing the apes and the other species. So it's not impossible to find differences. The big thing now is to really demonstrate that the thing that you find are indeed the basis of the trait you're interested in.
To test out their hypothesis, the researchers turned to lab-grown mice. So they began tinkering with this TBXT gene in the mice to see if they could change their tail length.
They basically try to modify the gene in the mice in the same way they are in the apes. And they showed that when you have a situation that is similar to that one that we have in the apes, even a mouse that usually has a long tail, it doesn't have a tail, or there is a variation in the length of the tail that you have.
Okay, let's pause for a second and digest that.
Yes, please.
When they played around with that gene in the mice, the researchers were able to shorten the length of the tail or remove the tail. But no matter what, when they started to play with that gene, the tail changed. But this isn't something new. Scientists play around with genetics in lab-grown mice all the time. But what sets this experiment apart is the sheer amount of laboratory data they collected.
That's what really makes a difference, is the amount of work they've done to really show that this change is enough to explain the trait. It's not that common. In fact, if you see around the comments of other colleagues about this kind of work, everyone is very impressed by the amount of work they've done.
So now that they've found out what gene is responsible for the tail or lack of tail, it's opened up a Pandora's box of questions. Do you have any questions right now?
I mean, you're explaining it so well. I guess so the work that they did with the mice in the labs, switching off the genes and seeing what happened to see if they could make them tailless like an ape. How did they track down that place where the gene is expressed? Tell me more.
Yeah, so they looked at a bunch of monkey and ape genomes. And that's how they were able to find this gene that was kind of different within apes and monkeys.
Okay. So first they recognized the pattern, and then they were like, hey, we have mice for this. We can switch this on and off and see what happens.
Yeah, mice were the field of experimentation, where they got to kind of test this theory about how our ancestors lost their tails in the present, which is also really cool because often when you're speculating about something that happened maybe 20 million years ago, you're not able to put together a live experiment.
Yeah, and it's just such a key trait for us apes to not have tails. So this is really, really, really cool.
Yeah, and the next big question is why? Why did we lose our tails?
The usual answer is, it's important for locomotion. But if you think about how humans, chimpanzee, gorilla, orangutan, and the others move around, you have all this, basically the spectrum. You have animals hanging from the branches with their arms. You've got animals working on the four legs. You've got animals working on two legs. So obviously it's not something important specifically for walking on two legs to be bipedal. So it must be something else. Or the event was just by chance. What is interesting here is that apparently when they did the mice with the mutation to mimic the situation in apes, they found that a lot of these mice did not survive, and some of them had problem with the development of the neural tube.
Okay, so your neural tube is developed in the first 4 weeks of pregnancy, and it's really important, and you've maybe never heard of it before because you don't go to the doctor and get your neural tube looked at, or you never hear about it. And that's because your neural tube is the precursor to your brain and spinal cord. It's what basically your entire nervous system comes from. And most fetuses that encounter a neural tube defect don't make it. This means any evolutionary increase in neural tube defects is incredibly risky. It's an incredibly high cost to pay for anything.
So that potentially in the ancestral old apes, for whatever reason, losing the tail increased the fitness. So it must have done something so important that is basically buffering the kind of risk, the kind of impact on the fitness of the animals. But if you're asking me why, there's a lot of speculation out there, a lot of interesting stories. But one thing is a story, one thing is an hypothesis that you can test. At least now we have the evidence from a molecular point of view that had happened. And sometimes you got things that are byproducts of other elements. So maybe not having a tail is a byproduct of the fact that this change changed the functionality of the gene in a way that is important for a thing we don't know yet. Maybe there is something else that this gene does in the form that is mutated in the apes that is important. And that's the reason why maybe this has been selected for. The thing here is that this phenotype is not human specific. But in our mind, is very much specific to human, because you look around, you see animals with tails all the time, and you just wonder, where is my tail?
He is delightful. And that is so interesting. I think it's just so neat that they're able to identify such an important trait through a single location on the genome. That's very, very interesting. I can see why he thought this was the most exciting discovery of the year.
Genetics is complex and often confusing, and to have something that is such a kind of a clear cut, you flip this switch and the lights go out, and you flip this switch and the lights go back on, it's not only rare, it's almost unheard of.
Okay, so that's number one. Who do you have for us next, and what is the discovery?
Our next guest is someone who you've heard before, Dr. Sofia Campercaro, who you may remember from last year's End of Year episode.
I definitely do. Oh, I'm so glad that she came back.
Yeah, me too. She's a zoo archaeologist and archaeological science lecturer at the Australian National University, and she's currently leading a project on Neanderthal sites in Spain.
So as you might guess, I'm really excited and interested in Neanderthals. So I found really exciting that this year they published what they call the Thorin, the last Neanderthal.
Before we get into the details of this discovery, it's important to mention that the finding of Thorin in southern France marked the first new Neanderthal fossil found in France in almost 40 years.
Oh, wow.
When Sophia told me this, my mind was blown, because I just assumed that with all we've learned from Neanderthals in the past couple decades, scientists must be finding Neanderthal bones all the time. But that's not the case.
We all know about Neanderthals, right? It's something that is in our popular culture. There are documentaries and movies every now and then come out, but there are actually not that many fossils available. So every time something new comes out, it's like, oh, finally, we can know more, because we are making interpretations based on the stone tools we find, because we associate them to what Neanderthals did, but we don't really have, a Neanderthal holding the stone tool. We haven't found that. But these ones in France, I think it's because they are taking the time and they have the opportunity of, take the time to excavate it properly and very slowly. I find it fascinating that it took 9 years to just excavate something like 50 to 30 centimeters.
Combined with how methodically they're excavating the site, the researchers have also been able to apply new methods and techniques, such as DNA analysis, which has transformed the mandible and collection of teeth that they've found into a genetic treasure trove.
It's, I would say, the most complete Neanderthal mandible and maxilla, so all the kind of teeth complex that we have. So thanks to that, they managed to get ancient DNA from the teeth, and that is telling us a lot about Neanderthal society, like the population structure. But they also managed to get very good dates on how old these fossils are.
And these fossils are between 52 and 42,000 years old, which puts them at the tail end of the Neanderthal life on the planet, hence the name Thorin the Last Neanderthal. The big surprise came when they compared DNA from Thorin to other Neanderthal DNA at nearby sites.
And when they did that, they found out that this Thorin, this individual, seems to belong to a different lineage, to a different population than what we thought. So for a long time, we always thought that Neanderthals were this big kind of family, right? They were from all the way from Siberia to Western Europe, to Spain, to Portugal, and they were all the same. Well, it's not the case now. Now we are seeing that actually, when they can look at the ancient DNA from these individuals, they can see there are kind of branches within this tree. And Neanderthals at one point, they kind of separated into different groups. In the case of this Thorin, it seemed that happened 100,000 years ago. So what it's telling us is Neanderthals were, kind of roaming around in Europe doing their thing. And then at some point, one of these groups kind of took off and stayed isolated for at least 50,000 years.
But they weren't geographically isolated. There were other groups nearby, But for some reason, they didn't interbreed.
Maybe they were like just like a grumpy group and they didn't want to, meet anyone else. They just were happy on their own small group. But there was no exchange of genes between other populations.
This is so wild. And it opens up a whole new line of research about the social dynamics between Neanderthal groups and how their dynamics might be connected to why they disappeared.
Yeah, that's what sprung to my mind, too.
It could be modern humans were just more social and Neanderthals were just grumpy people and they just didn't want to meet. And at the end, well, that was a population bottleneck. They just die off. So I think that's a very, it's opening a really interesting line of research of, okay, Neanderthals are not this monolithic culture that everything is the same. They were doing different things and they were probably different people. What we will never know is how different they look, probably not much, but it seems they didn't like each other, right? They had probably their own, social intrinsic relationships, and they have different population structures that we haven't been able to comprehend yet. If we consider that Neanderthal disappeared around 40,000 years ago, this must be one of the last ones. So they went with the idea of this is one of the last Neanderthals, one of the last Neanderthals of the mountain, one of the last kings of the mountain. Well, there you go, Thorin, right? So I think I really like that idea of putting these names that will stand out. So then we will remember these individuals. And I think it's also a way of showing they are not only bones.
I love it so much. She's right. The names matter a lot. And Thorin is a beautiful name. I mean, I love that we get to have a little bit of Lord of the Rings in our podcast.
Yeah, I agree. It's so evocative. And before she left, I asked her what this adds to our understanding of human evolution.
Every year, we are getting a new discovery that is showing us human evolution was not simple, and it's not this story that we studied in a school of. We have these Australopithecines, these apes, kind of primates in Africa, and then Neanderthals, and then modern humans. That's it. End of the story. Well, it's not. And it's building this really complex tree of evolution, if we want to call it. It's more like a jungle now. It's more like a mangrove than a tree. So genetics is bringing a lot to the discussion, because of course with bones it's really difficult to know. We all look the same without the skin, kind of thing. When you add genetics to it, it's when it's showing you the differences.
Yeah, she was so lovely. I'm so happy that she came back again this year to bring us a new discovery.
Me too.
Wow. Are you ready for our next discovery?
What do we have?
Our next guest is Dr. Fernando Villanea. He was on the show 2 years ago for our end of year episode. And he's a geneticist and a professor of anthropology at the University of Colorado in Boulder. And he studies human genetic ancestry to find Neanderthal and Denisovan DNA in people who are alive today.
Great. I remember Fernando very well and also had wanted him to come back on the show, so this is great.
Yeah, Fernando is great. And he brought us a story about saliva, or more accurately, salivary amylase, an enzyme that turns our mouths into an extension of our stomach.
Gross. Okay.
Yeah. All humans have multiple copies of this enzyme called AMY1, and the multiplication of this enzyme has long been associated with the rise of agriculture. But a study published in October found that this adaptation emerged 800,000 years ago, long before modern humans, Neanderthals, or Denisovans, who also had multiple copies of this enzyme.
So I chose to talk about the amylase duplication in hominids, and also the multiple duplications that have happened in modern humans because of agriculture. This is probably one of the first papers to come out of a new type of technology, which is called long-read sequencing. And I think we're going to be seeing a lot of new discoveries now that we have access to this technology. But this one specifically is about salivary amylase, which is the enzyme that helps us digest starch in your saliva. So in most mammals, they only have amylase in their pancreas. And so digestion of starch starts really late in the gut. But in primates, we have salivary amylase, right? So we pre-digest our starches, starting with the chewing in our mouth, and it really helps optimize getting those sugars out of starch. But the really like peculiar part of this discovery really has to do with this technology, right?
So, more traditional DNA sequencing technology use something called short reads. Our genomes are really long, and because our technology at the time couldn't fit all of it on the metaphorical page, we had to break them up into smaller fragments to get sequenced. Then a computer would take these fragments and assemble them back into a long sequence that includes your entire genome. But there were clear problems with short read sequencing.
The problem is when you have these duplications, right? These fragments that are identical, the computer will think they are just repeats of the same thing, right? They'll just like, there's like, oh, this is just like an error. There's like multiple copies of this thing. I'm just going to erase them. The long read sequencing allows you to read much longer, like thousands of base pairs at a single time. And so when you get a duplication in there, you know it's real. Like this is not an error. This is actually, there are two genes that are identical and they're next to each other. Previously, you couldn't see that. Now you can.
Are you following what all this means?
Uh, okay, so what I'm following is that we have an enzyme that helps us digest starches, and that's expressed in a very small section of genome. Is that?
Yeah. Well, okay.
Tell me.
What this means is that now when we read DNA sequences, like when we look at DNA that we have kind of unfurled, we're getting much more complete data because we get to see that things that are duplicates are actually duplicates. Like they are things that have been duplicated within our bodies for a purpose rather than just being like a clerical issue due to imperfect data recording. In this case, researchers using this long read sequencing technology noticed that there were a lot of duplicates of this salivary amylase enzyme in modern humans. And so instead of this being like a data issue, they could then start to look at why that is, why we have so many of these enzymes and what they do, rather than grappling with the question of, is our tool broken and why do we keep getting these duplicates? So it allows them to like ask better questions and also get clearer answers.
So what does this mean, and what do all these copies do for us?
Right. So anything that is in your genome that is a gene is going to be turned into protein, and that takes some amount of effort, right? There's like molecular machinery that needs to assemble. All these enzymes need to be put together. If you have multiple copies, that means that with a single assembling of your proteins, you can just make like 2 of these enzymes. If you have 17 copies, in a single run, you will make 17 of these enzymes. And that means that if that enzyme is doing something important, now it's doing it like 17 times faster. So in this case, like you are breaking down really long pieces of starch into really small sugars 17 times faster.
Okay, so let's step away for a second to process this.
Yes, please.
In the case of this amylase enzyme, the salivary amylase, having more copies isn't just a redundancy or protection against losing this enzyme, although it is that too. The more copies you have, the faster you digest starch. And so this is really important for our ancestors as they move from hunting and gathering to farming, which is essentially like growing as many starches as you can to eat. The other thing here is this form of evolution is a little bit different than how we usually think about evolution. Usually when we think of genetic evolution, we think of mutation. We think of a gene changing over time, like what we just talked about, the mutation of losing a tail, a light switching on or a light switching off. This isn't that. This is something called structural variation. And this in theory, is a much faster form of evolution because it doesn't require your genetic code to change. It doesn't require a mutation. And so instead of this salivary amylase enzyme changing, it just doubled or tripled or whatever the phrase is for 17 timesing. And this is bringing up some important questions when we think about our evolutionary history. Because we know that modern humans haven't been around that long. But we've expanded across the entire planet nonetheless. We eat a lot of different things, we live very different lifestyles, and yet we don't see that many genetic mutations.
The problem is we know that mutation is really rare, and it's so rare that when we see really like short times and we see really like innovative adaptations, it's unlikely that mutation is going to cause that just because it's so rare.
It does happen, though. The classic example is lactose tolerance in adults.
So basically, to continue to drink milk as an adult and not get some kind of gastric discomfort is due to a single mutation, which originates somewhere in human history, maybe some 10,000 years ago, but then becomes really common. And so a lot of us carry that mutation. And because of that, now that we have domesticated sort of cows and we can get milk and we can drink it, but that is almost the only example we have. And it's been really frustrating that nothing else we can really tie to a specific genetic change. And it's because mutation is so rare. But structural variation, this mechanism where a gene is duplicated and both duplicates now lay side by side, it's a different mechanism for adaptation. And as far as we can tell, that one happens a lot faster. So in this paper, they talk about how this amylase has been duplicated so many times in the last 10,000 years to the point where like some people carry like 17 copies of it.
And the explosion of amylase copies in modern humans is pretty recent.
Oh, so this explosion of all these copies seems to overlap with the beginnings of agriculture. So it seems like it's an adaptation maybe to having more starchy foods around.
Definitely. But it's not just modern humans that had that adaptation. In their study, they found that in 3 different Neanderthal genomes, they had between 2.6 and 5 copies. And in the one Denisovan genome they had access to, they found 8 copies. So our ancestors or cousins or however you want to frame them, our precursors, they were also eating starches and rapidly gaining the ability to process them faster.
Yes, and this is something I think that's been reflected in a lot of studies this year and in the most recent years about the wide variety of things that Neanderthals were actually eating. There's this misconception that we didn't evolve to eat starchy foods, but you can see it across Neanderthals and humans that that's not the case. So this is very cool.
Yeah, we were eating so much more than just meat. And now we can see that in our spit, literally.
Wow.
Another thing that Fernando mentioned was that sometimes these duplications are not good. And so I asked him, does this happen often? And he said that we should expect it to happen a lot, but they haven't found many examples yet, except...
I mean, we see it in immunity related genes a lot. It's the same kind of deal where like if you have more copies of our immune response, then you can tweak them and then you can have like a lot more adaptive response to pathogens. But that also has been frustrating to look at before we had this technology. And this is why I think the technology itself is also a big part of this discovery, is because we are going to start seeing a lot of papers about like adaptation for immunity. Like we're going to see a lot of papers for adaptation for other genes where we know that like more or less of these copies make a difference. I think about how like our species evolved in like a very specific environment for a very long time, and then in like really really fast we just kind of moved all over the place. And I mean that means that we had to have found solutions for all these problems, but I think we have yet to really crack like a lot of those. And so I'm excited to see what's going to come in the next few years.
as we all approach the holidays, having some knowledge that we're actually really well set up to process starch, I think might make us all feel a little bit better.
Yeah, if you're worried about it, just go into the holidays knowing that your body is adapted to our newly found post farming traditions.
For our next discovery, you've brought me something.
The discovery I brought today was just published on November 28th in the journal Science. And it's a paper called Footprint Evidence for Locomotor Diversity and Shared Habitats Among Pleistocene Hominins. But it's about fossil footprints. So this is a discovery by a large group of scientists, including many Leakey Foundation grantees, at a place called Kubifora in the Turkana Basin in Kenya. And this is a famous site where people have made many important discoveries over the years. And in this paper, the researchers talk about 4 trackways of hominin fossil footprints that they found there that dated to 1.5 million years old.
Wow.
Yeah. And so they found the footprints, they excavated them, but fossil footprints are really fragile, so you can't excavate them like you pick up a bone and take it to a museum. They'd fall apart. So they used modern technology and they 3D scanned the footprints and they analyzed them using something called photogrammetry. And they also compared them with other fossil tracks like the Laetoli footprints. And when they did that, they found that these tracks were left by 2 different species.
Whoa. So have they identified the 2 species?
Yes, they have. And they're 2 of my favorite ones. Homo erectus, which is likely an ancestor of ours, and Paranthropus boisei, which is a more distant relative that's different from Homo erectus in a lot of ways, including they found the way that these species moved and walked and stood. So this is what they think these 2 species are, and there's evidence to support it.
So why did you pick this discovery?
Well, for one, I love fossil footprints. They're so rare, but also they tell us things that bones can't tell us. And these footprints were left by these individuals at the same time, within days, hours, or maybe even moments of each other. And the researchers know this because they also did experiments around the shores of Lake Turkana, showing that the mud that can trap footprints or preserve footprints doesn't last very long before it's destroyed by waves or cracked by the sun.
Yeah, what else did they find in the mud there?
They found tracks from lots of other animals, like pelicans, antelope. They found zebras, hippos, elephants, and even giant storks. And they also found that the hominins were returning again and again to the habitat when other primates like baboons did not.
Wow. I mean, I can relate to that as somebody from the land of lakes. I love a good lake.
Absolutely. I mean, and lakes are great sources of food and water. So they think that maybe the Paranthropus boisei, because of the chemistry of its teeth, ate a lot of plants, grass-like, reed-like plants, which would have been plentiful around the lake margin habitat. And that there's also things that you can catch and eat. But it's also the interaction that's very cool to me.
Yeah. So what do you think this adds to our understanding of human evolution?
I think this adds further evidence that our story is not a single species story. It really shows, again, with concrete evidence that there were different kinds of us, equally adapted to the same environment at the same time in the same place, and that these 2 species repeatedly crossed paths with each other 1.5 million years ago and coexisted in this area for around 200,000 years.
Wow, that's incredible.
I think so too. And there's more to learn from these footprints. They'll continue to study them and hopefully find more.
I think this is such a cool story, and it makes me wish even more that there was a time machine, because we live in a time where we're the only one, and it's...
Yeah, it can get lonely.
thanks for bringing that discovery, Meredith.
You're so welcome. I was really happy to be able to bring you something and surprise you like you surprised me.
You did. Well, that's all that we have for the show today, but there were so many amazing discoveries that came out in 2024 that we couldn't fit into the episode.
Yes. So we have links in the show notes to articles about more amazing discoveries, and we have a newsletter that you can subscribe to. So look in your show notes for the links to both those things.
And subscribe.
Yes, subscribe.
It's always a joy, and I'm so grateful to have been on the show.
Thank you, Ray.
I will see you in the new year.
And before we go, I do want to give a special shout out to our editor, Audrey Quinn, for always helping us get the best out of our stories.
Thanks to Christian Capelli, Sofia Sampercaro, and Fernando Villanea for sharing their favorite discoveries of the year with us. And thank you to everyone who donated to support our show in 2024. We are so grateful for your generosity. If you haven't yet donated and would like to support Origin Stories and the Leakey Foundation, there's still time. And if you donate right now, your donation will be quadruple matched. So please go to leakeyfoundation.org/donate, or just click the link in your show notes.
Origin Stories is a project of the Leakey Foundation, a nonprofit dedicated to funding human origins research and sharing discoveries. This show is supported by our wonderful listeners with additional support from Jeannie Newman, the Ann and Gordon Getty Foundation, and the Joan and Arnold Travis Education Fund. This episode was produced by Ray Pang. Our editor is Audrey Quinn. Theme music by Henry Nagel. Additional music by Blue Dot Sessions and Lee Rosevere. Origin Stories will be back in 2025. Until then, we wish you a peaceful, healthy, and happy new year. Thanks for listening.